So it's a great pleasure to introduce to you Dr. Joaquin Espinosa. Dr. Espinosa is a professor at University of Colorado. He's also the director of the Linda Cernick Institute for Down Syndrome, which is the largest Down syndrome research center in the world. So I would like to thank everyone for the presence and Dr. Joaquin Espinosa for uh, accepting our invitation to give a talk today. So he's going to talk a little bit about the Linda Cernick Institute projects.
Bom dia. That's the last Portuguese you will hear from me today. There was a time when I spoke a little bit, and I used to grow up in Argentina, and I would come to southern Brazil a lot, and I lost it. So hopefully I'll recover some of it during the. Thank you, Ana Claudia and Bruna, for the invitation. It's been great so far, and I'm sure the rest will be great as well. And I'll present in English, but then during the Q&A, if you have a question in Portuguese and you speak it slowly, I should be able to understand it and reply as well. So as Bruna said, my name is Joaquin Espinosa, and I work for people with Down syndrome. One of my positions is being director of the Linda Cernick Institute for Down syndrome, and that is at the University of Colorado. And today we'll talk about some recent discoveries and therapeutic approaches in Down syndrome. And there are seats here in the front. Yeah. There you go. Let me know, Ana Claudia, if everything is good online, if I need to change anything about my behavior. Let's start slowly with some key concepts. I'd like to say that Down syndrome is the ultimate challenge in precision personalized medicine. Why? In precision medicine, the first step is discovering the genetic variant or the genetic cause of a particular type. We've known that in Down syndrome since the work of Gautier, Turpin, and Lujan, leading to the discovery that Down syndrome is caused by an extra copy of chromosome 21 or trisomy 21. Down syndrome is also, chromosome 21 was also one of the first chromosomes to be fully sequenced. So we know the genetic content of these chromosomes since the 2000s. We know there's no mutations which have an extra copy, so 50% extra genetic material. It's not dynamic. So the question is for us scientists, how we use this information to improve the lives of people with Down syndrome. Now, we should understand that Down syndrome is a growing population with many unmet needs. On the graph is a chart showing the increase in life expectancy. People with Down syndrome for different intervals, survival to 1 year to 5 years to 10 years. Look at what happened during the 20th century. Early in the 20th century, it was very uncommon to see Down syndrome live past their early childhood. But a baby born with Down syndrome in Sao Paulo today and receiving proper medical care, it would be expected to live up to 60 years old or so. So a remarkable increase in life expectancy. But then when the number of people with Down syndrome there on the right, and those are USA, again, broken by different age brackets, the different colors, you see that the fraction of newborns has been steady. But now because of the increased life expectancy, the total number of people has increased significantly. But not only the life expectancy has increased, and we're going to check. Yeah, I think that'll be better.
O outro eu tava ouvindo, Bruna. O de lapela não tava, não sei se tentaram o de lapela, mas ele não tá funcionando.
Yeah, and lower the volume a little bit.
Sim, agora eu ouço.
How about now? One,
Tá
two,
bom
three.
agora. Funcionando, Bruna, tá ok, obrigada.
Okay,
Thank
thank you.
you,
Yeah,
doctor.
this will be better. Thank you. So not only the life expectancy increased, but also their health, their quality of life has increased remarkably. And these are just some examples of individuals with Down syndrome that have become very popular in mainstream society because of their participation in, say, TV shows or movies or the fashion industry. And the point that I'm trying to make is that people with Down syndrome are here to stay. Right? Maybe the medical enterprise, maybe some aspects of society thought that with prenatal screening and the option of elective termination, that eventually the population will disappear. That's wrong. Okay, let's abandon that concept. There are more people with Down syndrome alive today than ever before, and there will be a lot of people with Down syndrome throughout the world making very important contributions to our society with their unique characteristics. Now, this is the first time that we have a sizable population of adults with Down syndrome. So it's a new phenomenon. And the epidemiology shows that they are remarkable in the sense that they have a different clinical risk profile. When you look at thousands of medical records of people with Down syndrome and you annotate the medical conditions that they have, you see that on one hand, they are protected from things like solid malignancies. This is not true for leukemias, but it's true for solid malignancies. Very rare to hear about a woman with Down syndrome having breast cancer, for example. I don't know of any. Very rare to hear an adult man with Down syndrome having prostate cancer, right? They're also protected from atherosclerosis and hypertension. I don't know of anyone with Down syndrome that died of a heart attack. Unknown, right? Also protected from allergies. Now, on the other hand, they're on the right, they're at a higher risk of many other medical conditions. They have a lot of autoimmunity. We'll talk more about that today. They have Alzheimer's disease. Pretty much all of them will develop Alzheimer's disease pathology about 20 to 30 years earlier than in the general population, with a lot of variation in the onset of dementia. They have a lot of complications from lung infections. We learned, for example, during COVID-19, that they are more likely to have severe symptoms and to be hospitalized. They have a number of vision and hearing issues and other conditions. So clearly, if we want to keep helping people with Down syndrome through biomedical research, we need to study the co-occurring conditions of Down syndrome. We need to study how the extra chromosome, in addition to causing these common but highly variable traits, how does the extra chromosome causes that increased risk of medical conditions? If we can figure that mechanism, we can help people with Down syndrome, but we will also help all of society. We will discover things that apply to all of us, right? So the question for scientists is, how come that an extra copy of chromosome 21 is modulating the severity and the appearance of basically all major medical conditions? Right? More specifically, which of the 200 or so genes on chromosome 21 are causing those effects? It's a manageable number, 200 genes. It's a manageable number. Of those 200, maybe 10, 20 will be the major driver of the phenotypes. So it's an insurmountable amount of work. How could we then counteract the undesired effects of gene triplication to benefit people with Down syndrome? Now, another concept that I think is key here is that everything that I told you so far is a generalization. That when you actually study a lot of people with Down syndrome, you realize that they're all dealing with the chromosome in their unique personal way. And that diversity is not a problem. That's an opportunity for discoveries. If you study enough people with Down syndrome, you will realize that although they all had the same genetic abnormality, there must be other factors that define which form of Down syndrome they present. There are individuals here like Sujit, Sujit plays 7 musical instruments in front of great, large audiences. He's clearly not impaired in his ability to learn and play music. Look at my friend Frank Stevens over there, who gave a very inspirational speech to the US Congress that launched a lot of new funding for Down syndrome research, right? Now, there may be other individuals here who are affected by many co-occurring conditions. They may have complex needs. Some of them may be non-speaking with a dual diagnosis of autism. So very clearly, two of them are the same. They are telling us that there are factors that modulate the impact of the extra chromosome. So what could be those factors? Well, it could be genetic variants, could be different alleles on genes on chromosome 21 itself. It could be alleles on other chromosomes that modify the impacts of chromosome 21. It could be epigenetic mechanisms. Check this out. You can have monozygotic twins with Down syndrome. So it's the same genome in the same womb. One of them having a congenital heart defect, the other one not. One of them being later diagnosed with autism, the other one not. So clearly, genes alone are not sufficient, right? Of course, there could be different phenotypes matched by different endotypes in the transcriptome, the proteome, the metabolome, the immune cell profile, the microbiome. Of course, there would be roles for environment, lifestyle, socioeconomic status, cultural factors. So clearly, given all the possibilities, we need an orchestrated effort that will help the community test a lot of different hypotheses. So everything that I told you was the intellectual fuel for the creation of the Linda Cernik Institute for Down Syndrome that started operations in 2012, 2013, and has now become the largest center for Down syndrome research in the world. More than 60 research teams, 200 scientists, many scientific publications. And you will appreciate that Down syndrome, being a multi-organ, multi-system condition, requires the involvement of many different disciplines. So sure enough, we have many departments across the university involved in these studies. And something that we're very proud of is that we work in close collaboration with the advocacy organizations, with the families, such as the Global Down Syndrome Foundation. One of the key studies in the Institute is this large cohort study of people with Down syndrome known as the Human Trisomy Project, or HTP for short. We have recruited more than 1,500 participants since 2016. And then once participants are recruited, they contribute the clinical histories, which we annotate to generate clinical metadata. We're big fans of multi-omics approaches, these large throughput, larger scale data sets. And then we're also big fans of open science approaches. We share all of our data sets through online portals that are accessible to the public, such as the Trisomy Explorer or the Include Data Hub. And this project is like an umbrella project that then has many nested studies, has supported many other projects, and led to many publications. Something that we learned is that people with Down syndrome love to participate in research. If you explain the research properly, the value of the proposition, they will come and they will give you blood samples, they will participate in complex research protocols. So thanks to the Human Trisomy Project, we've been able to analyze the impact of the trisomy on all these layers of biological information, right? In an unbiased fashion, without having to be too smart. Now with omics, we can ask everything at once and then the data, what is it that we need to focus on? So we've done all of these assays. We're big advocates, multidimensional biobanks. When you get a blood sample from somebody with Down syndrome, treat it like it is liquid gold, right? Nothing goes to waste. You take the plasma, the white blood cells, the red blood cells, take everything you can and you store it for many different types of assays. Today, I want to show you how this works. I'll give you one example of how a discovery from multi-omics data sets can lead to clinical trials very quickly, right? So this discovery was published in 2016, led to a clinical trial that started recruiting in 2020. That's not a lot of time, right? In 4 years, you can take a basic science discovery, turn it into a clinical trial. And that's the original paper, and many more that I'll tell you about. And that's the announcement of the clinical trial. So here's a key observation. People with Down syndrome are the largest human population with a genetic predisposition to multi-organ autoimmunity. Okay? This has a small sample size, 441 participants in the Human Trisom Project, deep annotation of their clinical records. More than 60% of adults with Down syndrome have been diagnosed with at least one autoimmune condition. The most common is autoimmune thyroid disease that manifests either as hyper or more likely hypothyroidism. Right? Then, this is interesting. We don't talk enough about this. More than 35% of adults with Down syndrome have been diagnosed with one or more autoimmune skin conditions, things like alopecia areata, atopic dermatitis, psoriasis, hidradenitis suppurativa, so on and so forth. About 10% of adults have been diagnosed with celiac disease. That's 10 times more than in the general population, right? A tenfold increase. There are other conditions also more common in Down syndrome. And then if you look at the overlap of those conditions in this pie chart, 25% of participants, we cannot tell that they have an autoimmune condition or not. 35% have one, 23% have two, 10% have three. And these are all conditions that get diagnosed during the pediatric age, the first 2 decades of life. So if you ignore for a moment that these people have an extra copy of chromosome 21, you were to study them, you'll be like, wow, this is multi-organ autoimmunity of pediatric onset syndrome. That's what you would call it, right? If you blinded yourself to the fact that they have Down syndrome, right? So we think that this is the tip of the iceberg. Clearly, the immune system is making mistake, it's attacking different tissues. Chances are there are other phenotypes or the traits of Down syndrome that are tied to this autoimmune profile. So this is the original publication that really caught our imagination. You know, we've been obsessed with this now for almost a decade. Trisomy 21 consistently activates the interferon response. So we use in this paper cell lines, primary blood samples. We did analysis of the transcriptome, the proteome, short herpin RNA screens, and in every data set, very clearly we saw activation of the interferon response. So what is the interferon response? It's a part of the innate immune system that we all have and we use to fight off viruses. When a cell is infected by a virus, that cell releases interferons, these small peptides, that then bind to receptors in neighboring cells and trigger JAK-STAT signaling and launch an immune response. So interferons are cytokines, of course, that activate many different immune cell types. We know that interferon hyperactivity is a risk factor for autoimmunity. When you do too much of the antiviral response, you are more likely to make a mistake and develop an autoimmune condition. So this is a zoom-in view of the pathway. There are receptors that get activated by the interferon ligand, and then upon dimerization, you see activation of these JAK kinases leading to phosphorylation and activation of the STAT transcription factors. And then you have a gene expression program that will lead to immune activation. This is how immune cells talk to each other. So why would people with Down syndrome have a stronger interferon response? Because 4 of the 6 interferon receptors are encoded on chromosome 21. There's a little piece of DNA on chromosome 21 where 4 of the receptors are encoded, right? There are 3 types of interferon signaling: type 1, type 2, type 3, beta ligands, beta and alpha ligands, gamma ligands, lambda ligands. The 2 type 1 receptors encode on chromosome 21. 1 each of the type 2 and type 3 receptors encode on chromosome 21. So now you have a human being that in every cell of their body, in every nucleated cell of their body, they have 50% more of these very important receptors for immune signaling. They have a supercharged immune system, okay? So now some data for the students here, some good old-fashioned Western blots, measuring protein levels from cells. These are skin fibroblasts from a euploid control donor and a person with trisomy 21. Nice dose response of interferon alpha, beta, and gamma. GAP-PDH, the loading control for the samples, and then MX1 and ISG15. These are interferon proteins or genes, right? This is how you and I induce these genes with increasing doses of interference. This is how people with Down syndrome do it. They start a higher amount, and then they overinduce these interference-stimulated proteins, right? I should point out that this hypersensitivity of trisomy 21 cells to interference was known since the 70s, right? But somehow it never caught hold, of the imagination of the field, right? Now we're going to go to more modern approaches. This is a large transcriptome study, 400 research participants, 304 with trisomy 21 versus 96 euploid controls. We take the entire blood, put it onto something that is called a PAX gene RNA tube, and extract all the RNAs. There's no tissue culture. There's no possibility of artifact. Then the volcano plot shows you genes that are upregulated to the right of the midline, downregulated to the left of the midline in Down syndrome. And then in green, we're color coding the genes on chromosome 21. So Lujan and colleagues were right. There is an extra copy of chromosome 21. So your chromosome 21 genes are up 1.54, right? And in the y-axis, you see the p-values, very significant changes. But look at all the black dots. These are genes on all the chromosomes that are clearly also upregulated or downregulated. So then you get the entire list, give it to a computer that's been trained to pick up pathways that are enriched or depleted in that signature, and look what the computer tells you. That the top activated, orange is activated, response in the blood of people with Down syndrome is the interferon gamma response. And look at who is there in number 3, interferon alpha response. And if you read this list, you will see many inflammatory immune signals also activated in the blood of people with Down syndrome. So if you look at individual genes, you see the receptors, of course, are up 1.5-fold. This is a log2 scale, by the way. If you see a 2-fold change, it's a 4-fold change. So the receptors, of course, are up because there's an extra copy of them. But all these other genes are interferon-stimulated genes, genes that we should only induce if we are fighting off a virus, significantly elevated. Look at those p-values in people with Down syndrome with a lot of inter-individual variability. So now you can take a collection of these interferon-stimulated genes and create an interferon score. This is used in the study of immune conditions such as lupus and other conditions, where you give every person one number, right? Interferon score. It's calculated from 16 different interferon-stimulated genes. This is the range of interferon activity in the general population, 96 control individuals, right? Hovering around 0. Look at people with Down syndrome. The mean difference, 13-fold. People with Down syndrome, on average, have 13 times more interferon activity than the general population. To give you a sense of the scale, we're running the same experiment here on people from the general population without and with COVID-19. Look at the scales. Scales are the same. People with Down syndrome are going around with an immune system that looks like the immune system of a person in the hospital fighting for COVID, fighting for their life with COVID-19. It's a huge amount of immune activity. Somehow, people with Down syndrome manage to go around with their lives despite this much immune activation, right? So then, in follow-up experiments, we documented the changes in the plasma proteome, and the title is very obvious, right? Trisomy 21 causes changes in the circulating proteome indicative of chronic autoinflammation, and these are the collaborators. So we use a proteomics platform called SomaScan. In this second version of the experiment, published later last year, we measure 5,000 proteins. And sure enough, there are proteins elevated in Down syndrome and proteins depleted in Down syndrome. We feed it to the computer. Look, it's up on top. Once again, the interferon gamma response, interferon alpha response, many of the inflammatory signatures. So now the signature is driven by proteins, not RNAs, okay? Many interferon-inducible proteins like beta-2 microglobulin. This is a protein that we use for antigen presentation. It's a protein that we use to tell the immune system about viral epitopes that we may be having in our cells. Look at LAG3, a lymphocyte activating protein. So then if you do a targeted proteomic analysis of inflammatory markers, you see massive elevation of inflammatory markers in syndrome. Cytokines like TSLP, IL-10, IP10, CRP, just a couple examples here. And again, log 2 scale, right? Many inflammatory markers elevated in Down syndrome. Now, among those, we have known pathogenic cytokines like TNF-alpha and IL-6. We know that elevated levels of these cytokines will give you autoimmunity because they are the targets of approved therapies used in autoinflammatory conditions. So TNF is blocked with TNF blockers. IL-6 is also commonly blocked for the treatment of many autoinflammatory conditions. These cytokines are elevated in Down syndrome, but more so if you have high interferon scores. So the higher your interferon score in the transcriptome, the more elevated your pathogenic cytokines, right? So it's not just RNAs going up. You are now producing inflammatory markers that will give you autoimmunity, okay? So then we look at the immune system and we use this mass cytometry technique, and I was happy to see some very powerful mass flow cytometers here in the building. It seems like you guys are doing quite a bit of immunology as well. The experiment reveals global immune remodeling with multi-lineage hypersensitivity to interferons in Down syndrome. So in summary, what we saw is that in addition to more interferon receptor and signaling, there are many cell types that are elevated in frequency in the blood of people with Down syndrome, and there are other cell types that are depleted. And overall, what you see is a tilting of the immune system towards a pro-inflammatory autoimmunity-prone base. Now, in the experiment, we put one antibody against one of the receptors and monitor the surface expression, the amount of receptor in the surface of the cells. There's 100 immune cell types here. Gray is the average signal for the receptor in the euploid controls. Green is in Down syndrome. In every cell type that we looked at, people with Down syndrome have more of the receptor, right? Stem cells, monocytes, dendritic cells, ILCs, B cells, T cells. Green is always to the right of gray. So the entire immune system of a person with Down syndrome is supercharged. These interferon receptors, this is true for the other receptors as well. So then what happens if your entire immune system is supercharged? Now, this is basic biochemistry. If you have more of the receptor, when you put the ligand, you have a stronger activity, right? Michaelis-Menten biochemistry 101. So we can measure that with antibodies to the activated STAT transcription factors, phospho-STAT1, STAT4, STAT2. This is how you and I do the vital response. We go from a resting state to an activated state, you know, with some variation from person to person. This is how people with Down syndrome do it. They overdo it. They overshoot. So they inhale a virus. When they launch the response, they overdo it. Right? They have more complications. They have more immune activation, more immune infiltration in the lungs. That's true for multiple cell types, for multiple STAT factors. These T-SNE plots, for the students that are doing immunology, summarize the major immune cell types in the blood of people with and without Down syndrome, and the clouds are color-coded for different cell types. Cloud number 1 is classical monocytes, cloud number 2, some form of CD4 T cells, so on and so forth. So in this T-SNE plot, we're color-coding by the impact of trisomy 21. Red are cell types elevated in Down syndrome, blue are cell types depleted in Down syndrome. And here we're color-coding by correlation in Down syndrome with interferon score. And this is one of those games of find the difference. These two plots look very similar, meaning cell types that are elevated in Down syndrome tend to be tracking with interferon score. Cells depleted in Down syndrome tend to track down with interferon score. For example, cloud number 6. Effector memory CD8 T cells. These are the commando cells of the immune system. These are cells loaded with weaponry that can come and attack a tumor, but can also give you autoimmunity. They're elevated in Down syndrome, more so if you have high interferon scores. Cloud number 9, non-classical monocytes. These are inflammatory monocytes that will contribute to the inflammatory response. Red in Down syndrome, but more so if you have high interferon scores, so on and so forth. An easier way to look at this is in this scatter plot. We have the fold change in Down syndrome versus the fold change per unit interferon score. Look at that line. Boom, right? The positive correlation. The higher your interferon score, the more change, the more dysregulated your immune system. And here's an example Again, those CD8 effector memory cells up in Down syndrome, but more so with high interferon scores. What happens if you activate your immune system a lot? You lose your naive cells. You lose the cells that haven't been activated yet. So you see a depletion of the T cells. Last paper before we change gears here. We did also plasma metabolomics experiment, trying to understand the metabolic deregulation in Down syndrome. Look at the title. Trisomy 21 activates the kynurenine pathway via increased dosage of interferon receptors. So this is a volcano plot of metabolites in the blood. Metabolites that are elevated and depleted in Down syndrome. At the top of the charts, the smallest p-value, something called quinolinic acid, which I never heard of before. Turns out that quinolinic acid is part of this kynurenine pathway of tryptophan catabolism. So tryptophan is an amino acid that we use for various things, like making protein. But under conditions of inflammation in an interferon-inducible pathway, this enzyme IDO1 will turn tryptophan into kynurenine, which can then become quinolinic acid. And both kynurenine and quinolinic acid are elevated in Down syndrome, right? Why is this meaningful? Quinolinic acid is known as the inescapable neurotoxin. It's a molecule that, if it gets to your brain, will poison and kill neurons that have NMDA receptors through a phenomenon known as excitatory toxicity. Memantine, which is an antagonist of NMDA receptors, protects from quinolinic acid toxicity, and in animal models of Down syndrome, memantine was shown to provide benefits, so much so that a Brazilian scientist, Alberto Costa, launched a clinical trial for memantine in Down syndrome, and Ana Claudia ran one of those sites here in Brazil. We know that the levels of quinolinic acid are associated with lower cognition in Alzheimer's disease. It's also a potent convulsant. If you get a rat and you give it a little bit of quinolinic acid, it goes into seizures, and we know that people with Down syndrome have more seizure disorders. So this could be a metabolic mechanism for neurotoxicity in Down syndrome, okay? So again, this could be explained by interferon induction of IDO1, the rate-limiting enzyme in the pathway. This is how you and I do IDO1 induction. This is control, that's interferon alpha. This is how people with Down syndrome do IDO1 induction. Look at that. They overblow it, right? So if you look at the blood of people with Down syndrome, they're making more IDO1, and the amount of kynurenine relative to tryptophan increases proportionally with the amount of interferon-inducible protein 10, a marker of interferon activity. So the higher your interferonopathy, the more of this neurotoxic compound you make, okay? So I could go on and on, but I want to pause here. I hope you are convinced by now that no matter what omics assay you do, you get an interferon signature on top, okay? So clearly, interferon signaling is hyperactive in Down syndrome. So what are the impacts of hyperactive interferon signaling in Down syndrome? So last year, we published a paper that took 6 years to put together. Look at the title of the paper. Replication of the interferon receptor locus contributes to hallmarks of Down syndrome in a mouse model. So what did we do here? We have a mouse model of Down syndrome called DP16 that has a segmental duplication of a fragment of chromosome 16. Chromosome 16 in the mouse has 120 genes that are on chromosome 21 in humans, including the 4 receptors. So that segmental duplication is similar to Down syndrome in the sense that it triplicates most of the chromosome, not all of it, but most of it. So then you have a mouse model of Down syndrome that has many of the phenotypes of Down syndrome. So what we did, we used CRISPR technology to delete the entire locus. We put a CRISPR guide there, another one there, and look at the genome sequencing. You read this chromosome 16, and then boom, you dip, and then you come back up again, right? So we cleanly deleted only the 4 receptors. So we have now a mouse that is knockout for the 4 interferon receptors. We cross with the DP16, and we get the $1 million mouse. DP16, wild type, the mouse that we generated, but check this out. Now we have a mouse that has a segmental duplication, but now it has only 2 copies of the receptor. All the other genes are triplicated. Now we can subtract the effect of interferon receptor triplication. right? And this is just the RNA-seq to show you that, yes, the receptors are overexpressed in DP16. Sorry, these are the receptors. These are other triplicated genes. But in our $1 million mouse that we call norm for normalized interferon receptor copy, now we go back to normal levels. All the genes in the triplicated region are unaffected, right? So a clean genetic. I'm amazed by the things we can do nowadays, right? In the lab with things like CRISPR, like we can come in and cleanly delete a small region of a triplicated region. So if you read the paper, you will see that having an extra copy of the receptors contributes to gene dysregulation in the mouse model across multiple tissues, contributes to lethal immune hypersensitivity. If you get the DP16 mice and you challenge it with a viral mimetic molecule, poly-IC, which is like a double-stranded RNA, so it tricks the immune system into thinking that there is a virus. There isn't. It's just a mimicker. The DP16 mice waste away and die. They cannot tolerate immune activation. But if you reduce copy number of the receptors, now they can tolerate it, right? So you rescue that immune hypersensitivity. Congenital heart malformations, half of these pups, just like half of newborns with Down syndrome, have a hole in the heart. The heart hasn't fully formed. We can improve heart formation by getting rid of one copy of the interferon receptor. We partially, not totally, partially rescue the developmental and cognitive deficits in these mouse models. And we also partially normalize the craniofacial abnormality in this mouse model. So with all this data, now we're ready to ask the question, what would be the therapeutic benefit of attenuating interferon signaling in humans with Down syndrome? So this is where we got very lucky, because the obvious question is, can drugs that lower interferon response benefit people with Down syndrome? And we got lucky in the sense that the pharmaceutical industry had developed a lot of medicines that can do this. Most prominent among them, this class of medicines known as JAK inhibitors. And this table is incomplete. It seems like every month or every semester there's a new JAK inhibitor approved for something or another. The point that I'm trying to make is that there are multiple JAK inhibitors made by different companies with different indications, right? A plus symbol says that medicine is approved for that indication. For example, this medicine, tofacitinib, is approved for rheumatoid arthritis, psoriatic arthritis, polyarticular juvenile arthritis, ulcerative colitis, and now other indication. So we got very lucky because we don't have to come out with a new drug. We need to see if we can repurpose a drug that already is approved, right? So here, we're not going to mess with interference. We're not going to mess with the receptors. What we're hoping to achieve is that with a small molecule, we can tone down the downstream step in the pathway, activation of the JAK-STAT signaling pathway. Right? Something that we really like about this approach, these JAK inhibitors are small molecules with a short half-life in the body. So if you take the half-life is 3.5 hours. So you take a pill now at 10 a.m. By 2 p.m., there's only half of it in your body. By 8 p.m., there's only 25%. Next day, it's gone. Right? So very clean pharmacology. You can interrupt the treatment if you need to. So how to design a trial? Now, this is where clinical trial design becomes very important. If you're going to ask families and patients with Down syndrome to come to you and put them on an experimental medicine, what's the incentive? So we realize that these autoimmune skin conditions, which are very prevalent in Down syndrome, are a big issue for the families and oftentimes are treatment refractory. They don't respond to other therapies. So we said, okay, let's do a bucket trial to treat 5 autoimmune skin conditions in one trial, alopecia areata, hidradenitis suppurativa, the balls. And that picture actually is a mild case of hidradenitis suppurativa. Sometimes these lesions are very, very large. Atopic dermatitis, psoriasis, vitiligo. As the participants to come in for 4 to 9 month treatments with one of these JAK inhibitors, which is FDA approved for the conditions, tofacitinib, also known as Gelshains. Now, here's the design. Ages 12 to 50, phase 2, single arm open label. They're all on the medicine. They know that they are on the medicine. 16 weeks with an optional extension arm. They must have moderate to severe skin conditions and one safety profile. So you may be asking why? I mean, these are FDA approved medicines. Well, but they haven't been tested in Down syndrome. So if you read the inclusion criteria of the clinical trials out there, it's almost impossible for a person with Down syndrome to participate Because you had to be so healthy, have only a skin condition. People with Down syndrome have other issues. So test safety again, determine the impact of those immune markers, define the impact on the skin pathology. Then why not, since we are doing this, take a look at cognition and quality of life, see if there are any effects there. So the trial started in 2020 and just finished activities, 40 participants. Let me say that again. In the thick of the COVID-19 pandemic, we asked people with Down syndrome who are at very high risk of COVID-19 pathology to come in to let us suppress the immune system. Who in their right mind will run that trial, right? Oh, because we knew going in that lowering the immune system may help them with COVID-19. And I know that in this institution, there are people who participated in clinical trials for JAK inhibition in COVID-19. And sure enough, JAK inhibitors are approved for COVID-19 because COVID-19 is not so much caused by a virus as it is caused by the immune response to that virus, right? So we did a trial, and what this chart illustrates is the medical complexity when you want to do a clinical trial in Down syndrome. These are the qualifying conditions. Half of them had alopecia areata, half had the boils, hidradenitis suppurativa, a couple had psoriasis. But look at all the other conditions that these participants had. So you have to be mindful of this when you set up your inclusion and exclusion criteria. Otherwise, nobody will qualify, right? You say, oh, nobody should have thyroid disease. Well, half of your participants don't qualify, right? Half had thyroid disease. Half had a congenital heart defect. 40% had obstructive sleep apnea. Many of them have obesity. So you had to be inclusive, and then you had to define safety protocols that can accommodate for these differences. So this is the safety data. It is safe. You can give a JAK inhibitor to somebody with Down syndrome just like you would give it to people in the general population. So I'm showing here exposure-adjusted event rates per 100 patient years. So every 100 patient years, if you get 100 people with Down syndrome and treat for a year with this medicine, you see 70 runny noses, 60 skin rashes, 30 coughs, 25 diarrheas. This is fine. This is what you would see, in this population. Now, without a placebo arm, it's impossible for us to know whether this is more or less than otherwise noted, but it is a safe intervention. Now, aim 2, impact of immune dysregulation. This is one of my favorite slides in the entire presentation. These are interference scores. I showed you this already in a different format. Interference scores are elevated in Down syndrome, median difference 13.7-fold, with a lot of inter-individual variability. When the clinical trial participants come into the trial, they're average for Down syndrome. Within 2 weeks, 8 weeks, 16 weeks of going to the medicine, look at the median differences. By 8 weeks, look at that number. We reversed the interferonopathy. They were 13-fold higher, now they are 13-fold lower. So we don't suppress the immune system, we normalize the immune system. Now you look at these two outliers. Piece of advice for the students. If you have outliers on the data, lean in, it's not a problem. There may be a lot of wisdom in those outliers. Let me show you the data now as a time course. Outlier number 1, AA2, she was coming down and then she had a spike, and then ended up the person with the lowest interference score. Outlier AA4 was coming down and then it shoot up. What do you know? AA2 got a COVID-19 vaccine 3 days before the blood draw. AA4 had a flu-like episode the week leading to the last blood draw. Right? So they preserve immune activity. They're on the medicine, but they preserve immune activity. So if they need to, they can launch an immune response. These are the cytokine scores. They are composed of the information on 4 cytokines: CRP, TNF-alpha, IL-6, interferon-inducible protein 10. Look at that number, up 3.27-fold. Within a few weeks of treatment, the JAK inhibitor down 3.11-fold. Again, no suppression, it's just normalization, toning down, including those very pathogenic cytokines, TNF-alpha and IL-6. TNF-alpha and IL-6 are elevated in Down syndrome. They go down when the participants are on the medicine. And this is important because doctors may say, well, I want to use a TNF blocker, or I want to use an IL-6 blocker. Well, if you use a JAK inhibitor, you get the effect of lowering TNF-alpha and IL-6, okay? This is fascinating. 70% of people with Down syndrome have auto-antibodies against thyroid peroxidase. This is an enzyme in the thyroid. So when you have auto-antibodies against that, you attack the thyroid, eventually you get thyroid disease. Look at that. We're reducing the amount of auto-antibodies. So these autoreactive B cells are being calmed down when you put the person on the JAK inhibitor. This is a second less common autoantibody against the thyroid, anti-thyroid globulin. So now we're asking, what are the autoantibodies maybe toned down when you put the person on the JAK inhibitor? These are the skin scores. There's a lot of data, but I'll show you some pictures. The point that I'm trying to make here, everything is going down. This is the investigator global assessment. Overall skin pathology improving. Lower score means better skin health. This is the families filling out questionnaires saying, yeah, the skin is getting better, lower scores. This is a tool to measure alopecia. This is a tool to measure hidradenitis suppurativa. And then the p-values start getting larger as we lose sample size, right? This is eczema and psoriasis. We didn't have as many participants. Now, this is not a placebo control arm. Who cares? You couldn't do a placebo control treatment when you are seeing these kinds of results. So this is a young man from Texas that came to Colorado, 17 years old, senior year in high school, a drum player, ready to prom, the big dance that they do at the end of high school. His hair is not growing. His CD8 T cells are attacking his hair follicle. He cannot grow hair. He goes on the medicine. Those CD8 T cells cool down. Now the hair follicle kicks in. And now we know why his friends call him Ed Sheeran. Because, he does have red hair, just like Ed Sheeran. I could go on and show you many, many of these pictures. Alopecia totalis. No hair anywhere, no eyebrow, no eyelashes, nothing. Right? Hair starts coming up. Another person. Sometimes you see rapid regrowth, sometimes it takes long. Man with psoriasis arthritis. Look at the skin lesions, the psoriasis, right? In the abdomen, in the legs. Look after treatment with the medicine. And then what you don't see here is that his joints got better. See, he uses a walker. Now mom says, oh, he's going on hikes. He's getting on the bike, to exercise. So much improved quality of life. How about cognition? So we didn't know what to look for. How would your brain improve if inflammation? So we measure everything. We measure working memory and executive function, overall development, expressive vocabulary, episodic and spatial memory, motor control, visual motor. We measure a lot of tests. Participants completed a lot of cognitive tests. And what we observe is self-reported improvements in positive affect, general cognitive function, and lower anxiety. And then in the CANTAB test, where we're measuring short-term memory, we saw improvements as well. But again, it's not a placebo control arm. So are the participants fooling themselves into a placebo effect? Is there a practice effect? We don't know. But let me tell you about this participant. One of my favorite participants in the trial. Young woman with history of Down syndrome regression disorder. I'll tell you in a minute what this is. When she came into the trial, we asked her to complete this test. It's a visual motor test, very simple. Draw a line following the path of the car or the motorcycle. And then we take the time and we measure the number of errors. She could not understand the task. In her own world, very difficult comprehension. She was obsessed with coloring the drawing. So it was a coloring exercise. While on the medicine, look at her performance, the improvement. Everybody could tell that she was doing better. Before joining the trial, she was receiving electroconvulsive therapy. She was wearing electrodes, stimulating her brain to get her out of that catatonic state. The benefits were so obvious that a neuroimmunologist prescribed the JAK inhibitor off-label and the medical insurance decided to pay for it. So what is regression? What is this regression disorder? This is a rare but devastating condition where a person with Down syndrome is doing great. They're going to school, they're living autonomously, maybe they're holding a job, and then boom, they crash. Something happens, maybe an immune trigger, where they go into a catatonic state, they lose their speech, they lose their personality, loss of ability to perform activities of daily living, hallucinations, delusions, aggression. So we think that a subset of these DSRD cases could be triggered by the autoimmune attack to the brain, something like autoimmune encephalitis in the general population, okay? So we documented several cases of getting these individuals back to the baseline with a JAK inhibitor, right? Imagine this, that young woman, 10 years of no memories. She doesn't remember anything between the ages of 21st and of 19 and 28, nothing. And now you talk to her like this, and she's back on it taking this JAK inhibitor, okay? So I wanna highlight this, the study was highlighting the cover of the Washington Post during Mother's Day this year, because it was the moms of these participants who really got the doctors to talk about this and to think about the possibility of an immune basis for regression. So this led to the first clinical trial for immune therapies in Down syndrome regression disorder in collaboration with Dr. Santoro at Children's Hospital, LA, shown here. So this is now the second trial. This is now is focused on regression, leave alone the skin, regression. 2 immune therapies, tofacitinib and intravenous immunoglobulin versus a non-immune medicine, a benzodiazepine that would help with the catatonia. Multi-site collaboration between the Shrinic Institute, Children's Hospital Colorado, Children's Hospital Los. So we finished the first trial. We're about halfway or so, the second trial, and now we're about to start the third trial. The third trial will be in the pediatric population. It will be a phase 2 to B3, pediatric population, kids with Down syndrome, with no co-occurring conditions or with whatever co-occurring conditions they happen to have. We're not gonna select, right? And 6 to 12 months of JAK inhibition in the phase 2 would be 6 months, later would be 12 months. When we get to the phase 3, it will be randomized placebo control, inclusion-exclusion criteria. As in pediatric rheumatology, we would pretend that these kids have juvenile arthritis. That's what we would do. We'll forget that they have Down syndrome, juvenile arthritis. You qualify for this medicine, let's give it to you, and then see what happens, right? And 1, safety profile and clinical benefits through adverse event monitoring, health assessments. And 2, improvements in cognition, language, social development, neurological health. And 3, all the multi-omics to look at what happens at the biology of Down syndrome when these kids go on medicine. So in conclusion, interferon hyperactivity causes many health issues in Down syndrome, such as autoimmune disorders, but also severe complications from non-viral infections. Research into immune system dysregulation has led to first-in-kind clinical trials for immune therapies, such as JAK inhibitors in these populations. And we strongly believe that restoring immune balance could have many benefits for people with Down syndrome, even perhaps from early development. So we like to, one of the things that we say is we like to understand Down syndrome in part as an interferonopathy. Interferonopathy is a thing, is a group of monogenic neurodevelopmental disorders called type 1 interferonopathies caused by too much interferon signaling. Now, before I wrap up, I want to share with you some amazing news that are relevant to this institution. Last year, the NIH, the National Institutes of Health of the USA, announced the creation of the Include Down Syndrome Cohort Program of the SCDP. What is this? This is meant to be the largest, most detailed study of people with Down syndrome today, involving Down syndrome cohort research sites, many sites, that will develop a common protocol under the guidance of our clinical coordinating center that will engage the DS community. They will send biological samples to a biorepository, and all the data eventually will be contributed to a data coordinating center and shared back with the community. So we apply to be part of this ambitious research project to create the Human Trisomy Project Latin American Network, to make sure that people with Down syndrome in Latin America are represented in this study. Why? Because of many good reasons. First of all, from a USA-centric perspective, Latinos are the fastest growing segment of the population with Down syndrome in the USA, right? But second, because we think that Latinos with Down syndrome can be very informative, especially if you include the full representation across the Latin American continent. Latinos, we're not a monolithic group, right? It's very different from one country to another, not only in ethnic composition, but our diet, our culture, our favorite soccer teams. You know, there are many things that are different. So chances are those differences modify how Down syndrome presents. So the goal is to recruit 1,500 Latinos from the USA, Mexico, Colombia, Brazil, Argentina, Chile, plus satellites, pilots in Bolivia and Venezuela, 250 participants per each of the main sites. So yes, 250 participants recruited here at this hospital, 100% Latinos, all comers, clinical data, cognition, imaging, cardiometabolic, multiomics, whatever the common protocol demands. So we were super, super happy that we were awarded this grant. We got the money. We're working on the common protocol, and Ana Claudia and Bruna will be leading the activities here for the Brazil site. So we'll start with Latin America first, and now we're already in motion to achieve similar efforts in Asia, Africa, Australia, to make sure that all people with Down syndrome are well represented. Last but not least, there's like 50 or so scientists and support staff at the Synchronic Institute. If I put the names here, you won't be able to read it. So I'm gonna restrain myself to acknowledging the key program leaders at the Synchronic Institute, Kelly, Angela, Matthew, and Lindy, funding from the NIH, but also from the Global Down Syndrome Foundation and the Anna John JC Foundation. Many wonderful collaborations. And as I mentioned earlier, very important to be working in close collaboration with the advocacy organizations, in our case, it's the Global Foundation. And with that, I hope that we have questions and criticisms. Thank you, everybody. Questions in English, in Spanish, or Portuguese?
Yeah, we're open for questions. English, Portuguese, Spanish.
There's one here in the front, Bruno.
Yes.
Great talk. I'd like to know whether this overproduction of interferon and interferon pathway would affect the development of embryo and the fetus of these people with the trisomy.
This is a great question. The question is whether the interferonopathy can affect embryonic development. And the answer is yes. In the mouse model, some of the phenotypes that were rescued when you normalize copy number of the receptor from 3 to 2 are embryonic phenotypes, like the congenital heart defect, right? So we measure that at birth, right? So the heart is having a hard time growing well in those embryos, and it's unlikely to form the septums, right? And then you have these little holes, and you can have an atrial septum defect or a ventricular septum defect. When the embryo has the normal copy of interferon receptors, the heart forms well. The other phenotype is a craniofacial abnormalities. That's an embryonic phenotype, right? That starts craniofacial malformation, it starts in the embryo. So I actually removed some slides that were about this in the interest of time. If you take the embryo, the mouse embryo, and you do the transcriptome analysis, you see the interferon signaling in the heart, in the brain, in other tissues. But now we have data from a biobank in the USA that has single-cell RNA-seq data on human embryos. And we see the interferon signature in the heart, in the brain, and in other tissues. So if you take this farther, so well, how could we, let's assume that our trials go well. How could we start even earlier? Can we give a mom who is pregnant with trisomy 21 a JAK inhibitor? Well, you buy a JAK inhibitor and it says, do not take if pregnant. But every medicine has that, right? Why? Because nobody knows what will happen. So it's not because it's known to be bad, it's because there's no knowledge. But it turns out that women with psoriatic arthritis, ulcerative colitis, they have sex and get pregnant. They've been told by the doctor, careful, let me know if you get pregnant. But by the time they know they're pregnant, maybe 2-4 months have gone by. And they come to the doctor and say, what should we do? So now there are many meta-analyses in the literature, in the rheumatology literature, that says, we don't see any ill effects, it seems like. So then the doctors had to work with the mom because the mom has arthritis. If she comes off the medicine, the arthritis will come back. So the rheumatologists are having this conversation about what to do with a pregnant woman, that has an approved indication for the JAK inhibitor. So far, the data says there's no issues with taking a JAK inhibitor while pregnant. But the other great implication of your great question is that immune activation in the moms may have an impact on the embryo because the embryo is very sensitive to immune activation. And we know that immune activation in the moms can affect embryonic development, such in the TORCH syndrome, which I know how it translates in Portuguese. So if the mom has an infection, some infections, Bordetella, other things, can impact the embryo. And then Zika virus. The Zika virus is a wonderful experiment where in a mouse model of Zika virus, if you delete the interferon receptor encoded on chromosome 21, interferon alpha receptor 1, the embryo is not affected as much. Again, it's the immune response that affects the embryo, not so much the virus itself. Of course, the virus causes the immune response. So you're absolutely right. We need to think about the prenatal embryonic effects of this interferonopathy.
Hello, this was great talk, amazing.
Thank you.
I'd like to know more if you're pursuing these gene editing strategies to go for preclinical stages, and if you thought about instead of deleting, using like epigenome editing.
You say genetic strategies? Is that
Gene editing like CRISPR?
Yeah. Well, we're not. There's some investigators that are using chromosome silencing. How in females, one of the copies of chromosome X is inactivated. It doesn't express the genes. That's done through this epigenetic silencing through the exist locus. So there's a way of getting an exist locus engineered into chromosome 21 and silence one copy. That can be done in vitro in cells that have been done successfully. To a degree, it can be done in mice. But it's very hard to envision how that could be done in a whole human, right? How would you silence one copy of chromosome 21 in every tissue? It gets you, back to the same difficulties of gene therapy, right? Is that what's your vector going to be? How are you going to ensure no off-site effects or off-target effects? I think that given everything that I told about the immune system, what is more reasonable, perhaps feasible, is to normalize the immune system, right? You don't have to change the entire body. Maybe you can edit the immune system like you would do in sickle cell anemia, like you would do during a bone marrow transplant, to give people with Down syndrome an immune system that has only 2 copies of the receptors, only 2 copies of the chromosome. And that may have benefits. Chances are that there are people with Down syndrome out there that have a euploid immune system. This would be people with Down syndrome who had leukemia, and the treatment was bone marrow transplant, where their bone marrow was wiped out with radiation, and they were given a new bone marrow from a euploid donor. So chances are there's a group of people with Down syndrome out there walking around with a euploid immune system, and I would love to meet them and learn from them. CRISPR, again, very hard. How do you CRISPR out one copy of chromosome 21 in every cell of a fully developed baby? I think technologically we're not there. But I think in terms of doing it in cells and in mouse models could be very informative to learn the mechanistic aspects of the trisomy.
And there are a few new papers like using CAR cells against autoimmune disease like lupus and fibromyalgia. Do you think it could be also a strategy for Down syndrome, thinking like as autoimmunity disease?
That's a very interesting proposition. So the question is about using engineer T cells, with chimeric receptors to address some of the features of Down syndrome. There's one obvious application of that, which is in the leukemias of Down syndrome. I didn't talk much about that, but some leukemias are more common in Down syndrome, like B-cell acute lymphocytic leukemia or acute myeloid leukemia. And the B-cell leukemia of Down syndrome has a very common genetic abnormality, is rearrangement of the so-called CRLF2 receptor. In typical B-cell ALL, this is only 5–10%, I think. In Down syndrome, it's more than 50%. So if you do a CAR T therapy attacking, cells that have an extra, that do a lot of this receptor, that'll be a great strategy for leukemia in Down syndrome. And that is being developed by experts in immunotherapy in the USA. A CRLF2 targeted CAR T cell for B-cell leukemia and Down syndrome. For other applications, I don't know. Well, maybe you're right for some of the autoimmune conditions. There could be a CAR T therapy that could be devised.
Just one last question.
Please.
More like a curiosity that when I saw your data about the transcriptome, I saw there was also upregulation of the gamma globins. Actually, we work here with sickle cell anemia. So I was just curious if anyone observed that this actually results in a high expression of fetal hemoglobin.
Ooh, that's very interesting. So let's see if I can find this slide. Towards the beginning. So I spent a good chunk of your morning telling you about interferon, but look who is there at number 2. Heme metabolism. The second signature in the transcriptome of people with Down syndrome. What is this? When you unpack that signature, it's a signature of increased erythropoiesis, red blood cell formation. And it's also a signature of hypoxia inducible genes. And in that signature, you see gamma fetal hemoglobins. What is this? Look at the blood of people with Down syndrome and you put it under the microscope. The first thing you see is they have fewer, bigger red blood cells. It's called microcytosis. It's a sign of new red blood cells. Right? If you measure erythropoietin, this is the key factor driving red blood cell formation. This is the stuff that Lance Armstrong injected himself, to increase his respiratory capacity and win the Tour de France. They have a lot more. It seems like people with Down syndrome are in a chronic state of hypoxia, molecularly speaking, right? They're making red blood cells all the time. They make more erythropoietin. And they may be doing, or they're very likely to be doing what's called a stress erythropoiesis, meaning making red blood cells outside of the bone marrow in places like the spleen or the liver. And that's when you activate the gamma globulins, right? When you go into a stress erythropoiesis to make more red blood cells because you are not meeting the demand, you're likely to go back into the developmental hemoglobin program and start doing the gamma fetal hemoglobins. So we see this also in the mouse model side. We published that this year, an entire paper on the investigation of the heme metabolic signature. Where is the hypoxia coming from? It could be the obstructive sleep apnea. They have a lot of obstructive sleep apnea. More than half of adults have issues at night, where maybe because of the hypotonia or differences in the upper respiratory airway, they suffocate at night, right? And then that'll give you hypoxic events. Could be that. Could be poor heart function, right? The heart, although it doesn't get atherosclerosis, it doesn't get occluded. Maybe it's not, pumping enough to meet the demand. It could be other things. It could be a lung maturation issue. But very clearly, the red blood cells of people with Down syndrome are different. Yeah.
Thank you very much.
Just a follow up on the question. Since you mentioned the
Hypoxia?
hypoxia,
Yeah.
Have you looked at your participants from Denver against
Yeah.
people outside from Denver?
And it's not that different. It's a good question because many of our participants live in Denver, Colorado, which is about 1.5 kilometers above sea level. So you may think, well, maybe that's why you see the hypoxia signature. It's true for participants in New Orleans at sea level. So we didn't see a big difference there.
Hi, very nice talk. I would like to know about the cognitive function phenotype that you analyzed. Did you analyze anything about autism symptoms, or could you see any relation regarding the immune activation and autism symptoms?
Great. Also relevant. You know, autism spectrum disorder, we think is more frequent in Down syndrome. And the numbers change depending on the study. And also, there are some difficulties in diagnosing autism in Down syndrome. But the numbers range between 10 and 20%. That's a large fraction of the population. And my question to people who know better, to people maybe like you who understand autism is, how will you tell autism apart from regression? And how much of that autism is a very early episode of regression when the kid was young, where you didn't get to see the delta, the drop, because the person never fully developed. I kid you not, I have an email in my inbox from a mom, who's also a doctor, yesterday, telling me, Joaquin, based on what I see on my daughter who is receiving an immune therapy for her regression, I'm seeing things that we thought were part of the autism phenotype. She's having social interactions, she's having social reactions that she shouldn't have because of her autism diagnosis. So I think that autism, not me, the mom is saying in the email, I think that autism and regression may be very similar, that the autism may be a regressive phenomenon, and it happened very early. You never got to see what that person was capable of, because the person started developing and then went back to that. So I just forwarded that email this morning to the pediatric neurologist that I work with, being like, hey, how much of autism could be a regressive episode? Should we think of these immune therapies for what we're calling autism, right? Let's see what they reply, right? But I think that you're right on the money. And if you read the autism literature, there's a fraction, autism is a spectrum, of course, there's a fraction of autism that have clear immunotology. For example, there's a fraction of autism that are associated with autoantibodies in the mom, right? Where the mom seems to have an autoimmune profile, and then some autoantibody seems to be, going through the placenta and affecting the embryo. There's literature showing that some of the cytokines elevated in Down syndrome, like IL-17, in mouse models, if you have that immune activation in the mom, IL-17 can then confer autistic features to that embryo. So I think that we should think about a potential immunotology for some of these cases of autism in Down syndrome, absolutely. And we do measure, autistic features, through the social, through the language and social communication tools. It's not a full workup for autism, but it contains some of the elements that will capture changes in autistic phenotypes. Yeah.
One question.
Great talk.
Great talk, thank you. Are there other immunotherapies targeting the autoimmune disease that you include in the first trial? Because I'm wondering if could be interesting to compare the effect of other immunotherapies in comparison with the ones that are targeting the JAK-STAT, because then you could show that something that makes sense biologically actually will change the indication of the therapy, and showing that the JAK-STAT pathway actually makes a huge difference. Because my main doubt is that if you use another advanced immunotherapy approach, you would have a similar effect, even if it's not what we would expect biologically.
Thank you. Great question. Yeah. I show you a lot of data, a lot of immune pathways, a lot of cytokine stimulators. So why a JAK1 inhibitor is our preferred choice, right? In our current understanding of immune dysregulation in Down syndrome, it all starts here. It all starts with an extra copy of these interferon receptors, leading to higher baseline, but also higher induction of the interferon response. And all three, it's not shown here, but all three types of interferon signaling, which are elevated in Down syndrome, they all use the JAK1 kinase. And it can be in combination with another kinase. It can be JAK1, JAK2, JAK1, TYK2. So if you have a molecule that blocks JAK1, we think that will take care of the root of the problem, right? So we think that's the most parsimonious, the most elegant solution. Now you could say, well, maybe you could block the interference, and there are ways of doing that. Maybe you can use monoclonal antibodies against the receptors to block that. Maybe you want to go farther downstream and block IL-6 or TNF-alpha. So clearly there is a menu of possibilities. In our understanding of the science, none of those options will be as elegant as going after JAK1, right? Because either they won't address all the issue, right? So if we use say a TYK2 inhibitor that will block this pathway but not the other two. If we use an antibody against the receptors, we have 4 receptors. Which one are you going to block, right? And if you go downstream, you say, well, I should block IL-6, and some people are very adamant about that. How about TNF-alpha? So now you run into a problem of multiplicity and redundancy. So we think that first we need to define the effects of JAK1 inhibition. Now, with all that being said, I think we do need to explore other strategies. And if you ask me what the second priority should be, I would think it would be none of the biologicals that pharma is manufacturing with a price tag of $6,000 a month. In my mind, the next step should be natural compounds with anti-inflammatory properties. In my mind, we need the native populations of the Amazonian basin to tell us what are the compounds made by trees in the Amazon that have an anti-inflammatory property equivalent or partially equivalent to a manufactured JAK inhibitor, right? So if I had the resources and the bandwidth and the time of my life, the clinical trial would be JAK1 inhibitor, which I show you normalizes all these markers right next to curcumin, apigenin, green tea extract, something out of the Amazon rainforest, you tell me. So there has to be a natural compound that would achieve these effects, even if partially. And then, we can formulate it, provide it as a supplement with a great safety profile and make it accessible worldwide. But your point is well taken. We need to look beyond the JAK1 inhibitors.
So I would like to thank Dr. Joaquin Espinosa and everyone for the attendance. Thank you.
Pode ficar. Vai ficar.